Hi, everybody. We are going to do some work with Punnett squares today. So you should have already watched the Amoeba Sisters video introducing Punnett squares. One reason I like to have them kick it off is because they do a nice job of really using all that vocab that we learned to objectives before. So all of that genetics vocab, heterozygous, homozygous, genotype, phenotype, hybrid, purebred, dominant, recessive. Now we're going to put all that into use when we talk about predicting genetic crosses, their outcomes with Punnett squares. So objective number 7 is I can use Punnett squares to predict examples of new genetic combinations that are the result of sexual reproduction. So just wanted to remind you, we skipped the essay for number 6, so we're moving on to number 7. So I wanted to kind of go through a couple practice problems with you guys. And what you guys will do, this is a Canvas Studio assignment. So as I'm recording this along the way, you guys will see some locations where you have to stop and answer a question. So I'm going to ask you to predict what would go in the boxes of the Punnett squares, and you have to answer correctly in order to be able to move on with the video. Just an FYI, if you answer incorrectly, then the video will take you back to the last question, and you have to rewatch that portion of the video again. All right, so we're going to look at the first example. So a Punnett square is simply a genetic tool. We are going to be using what's called monohybrid crosses, crossing and following just one trait at a time. Next year when you get to high school, you'll do some dihybrid and trihybrid crosses where you follow like 2 and 3 characteristics at a time, but we're just going to go on with one characteristic. So every time you complete a Punnett square, you are just going to need to draw a simple 2x2 block, just like here. So our problem for this Punnett square is a green pea plant, and it gives us in parentheses capital G, capital G. That is that plant's genotype. They have 2 dominant alleles. So a green pea plant is being crossed with a green pea plant. Now notice the second green pea plant is heterozygous dominant. They have a capital G representing a dominant allele, but then also a lowercase g in their genotype, and that represents the recessive allele. So notice the phenotypes, both plants are green. One is homozygous dominant, capital G, and the other is heterozygous dominant, capital G, lowercase g. So just got to know that vocab. So when you complete the Punnett square, you simply take one parent's alleles, you split them apart and put them on top of separate boxes on top of the Punnett square. And then you take the second parent's alleles and you do the same thing. You split them apart and you put them to the left of the 2 boxes on the left. Now, you might not realize it, but that splitting them apart is representing Mendel's law of segregation. Remember, Mendel said that gene pairs separate during meiosis. So what a Punnett square is basically showing us is this parent, half of their gametes have a dominant allele, and half of their gametes have a recessive allele. And this parent, because they are homozygous, all of their gametes have a dominant allele. But what a Punnett square shows us is the possible genotypes of their offspring. Now, a Punnett square does not say these 2 plants are going to be crossed and they're going to have exactly 4 offspring, and the 4 offspring are going to have these exact genotypes and phenotypes. It's a probability, and that's what I like about the end of that Amoeba Sisters video. She talks about how it just shows you that the chances, but we all know families that have like 4 boys or 4 girls. So the genetic probability repeats every time fertilization occurs, and a Punnett square shows the probability of what the offspring will look like, their genotypes and phenotypes. So once you set up your Punnett square by separating the alleles and putting them on the top and on the left, then you fill in the 4 boxes. So you grab the allele from the top, you copy it in the top left box. You grab the allele from the left, you copy it in here. You move over to the next box, capital G here, capital G here. You move down to the second row, capital G from the top, lowercase g from the left. You move to the last box, capital G from the top, lowercase g from the left. So once you have filled out all 4 boxes, then the last thing you have to do is you have to interpret the results. And this is a step that gets left off a lot. People are like, sweet, I filled out a Punnett square, great. That doesn't tell me that you know anything. You need to now interpret what that means. So A genotypic ratio is often asked for, and in this case, they have a 2 homozygous dominant to 2 heterozygous dominant to 0 probability of having a homozygous recessive offspring. And then the ratio for the phenotypes, this, and this all results in a green pea plant. So they have a 4 to 0 chance of green pea plants, or 100%. So we use ratios, but we also use percentages to interpret our results for Punnett squares. And unless it specifically says, it doesn't matter to me which one you use. If you said 100% or 4 out of 4 are going to be green. So we're going to practice some together. I have another problem for you. We're going to set this up. We're going to fill in the blanks, and then we are going to interpret the results. So a green pea plant is crossed with a yellow pea plant. Now, notice I gave you the genotypes. You're going to start seeing all sorts of different types of Punnett square problems. We have a couple of examples of different types in this video. Sometimes the genotype is given to you, but sometimes it uses the big words, a heterozygous dominant green pea plant. And you have to interpret, okay, if it's dominant and heterozygous and it's green, green is the dominant phenotype. So get used to applying that vocab and reading the clues in the question. So it doesn't matter which parent you put on the top and which parent you put on the left, but we're going to separate this genotype right here. So I'm going to put a capital G here, and I'm going to put a lowercase g here. And I'm going to move over to the left. And now this yellow pea plant, I'm going to separate their gene pairs. I'm going to put a lowercase g there and a lowercase g there. And I did use blue and pink to represent the father and the mother's contribution, just getting the idea. Now these are plants, they are not boy or girl plants. They have both parts, but that's just wanted to kind of point out to you. It doesn't matter if you put like mom on top or dad on the other side. It doesn't really matter here. You can set it up either way. The results will be the same. So now we're going to go ahead and fill in this top left square. So we want to grab, once again, the allele from the top, which is a capital G, and the allele from the left, which is a lowercase recessive or yellow allele. I'm going to move over to this box. On the top, I have a lowercase g, and to the left, I have a lowercase g. Down to this square, on the top, I have a capital G, and to the left, I have a lowercase g. And then this final box here, on the top, I have a lowercase g, and to the left, I have a lowercase g. So we filled out our Punnett square, yay, but we now need to interpret the results. So our genotypes here, we end up with, and if we're going to use ratios, we would end up with 2 heterozygous dominant to every 2 homozygous recessive. And then if we're going to interpret phenotypes, this and this are going to result in a green pea plant, and this and this are going to result in a yellow pea plant. So our phenotypes there are going to be 50, we'll use percentages now just to get used to that, 50% green, and then we have 50% yellow. So we've just completed and interpreted our first Punnett square. Let's go ahead and set up another one. So we have a tall plant is crossed with a tall plant. Now if we look at the genotypes, this tall plant is homozygous, this tall plant is heterozygous. We have to know, because their phenotype is tall and they have dominant alleles, that tall is the dominant allele. So to set this one up, I'm going to put over here this time, I'll put capital T, and right here I'll do another capital T. We've taken this genotype and put it on the left. Now this tall parent plant, I'm going to put up here, capital T, lowercase t. Now we're going to fill out each square. So in this box, we copy the allele from the top and the allele from the left, and that gives us a genotype possible, capital T, capital T. In this box, allele from the top, allele from the left, capital T, lowercase t. This box, allele from the top, allele from the left, capital, capital. And in this box, allele from the top and allele from the left, capital T, lowercase t. So the genotypic ratios for this cross, we get 2 homozygous dominant parents to every 2 heterozygous dominant parents. Our phenotypes, however, if you notice, the only possible phenotype is tall plants. Okay, so these are all going to be tall, so we could say 100% tall. You could also say all of them are going to be tall, and I'm A-okay with that. All right, example number 3. These are going to get a little bit more wordy. We're going to try and challenge you. So the trait of tongue rolling, we're going to use a capital T to represent tongue rolling, is dominant over the trait of not tongue rolling. Lowercase t there. So complete the Punnett square below to show a cross between, says and. Should probably have that say a homozygous tongue roller and a non-tongue roller. Now, I hope you know what I'm talking about. Tongue rolling is actually a genetic inherited trait. I'm going to go very cautiously Google this and see if we can find it. So, tongue rolling genetics. So, this trait right here, this is what I'm talking about. So, I'm going to come put this in my PowerPoint, show you what I mean. I can't do that, or I would have showed you how to do it. Now, you might be thinking, well, Mrs. Murray, you just haven't taught yourself to do that yet. Nope, I've tried. I've stood in front of several thousand eighth graders teaching this for 13 years and tried to roll my tongue like that, and I cannot do it. You want to know why? I don't have a gene for it. So, believe it or not, you inherit this characteristic. So, if we are going to cross here, a homozygous tongue roller. So if they can roll their tongue, that means they have to have a dominant allele. So I'm going to put that parent up here. They're homozygous. Their 2 alleles are the same. And a non-tongue roller, that's me. So non-tongue rolling is recessive. So I'm going to do lowercase t here and lowercase t here. So fill in the Punnett square. Grab the allele from the top, the allele from the left. Allele from the top, allele from the left. Allele from the top, allele from the left, allele from the top, allele from the left. And when we do that, we see that these parents, all of their offspring have the same genotype, no matter what. They have a 100% chance. So 100% of their offspring are heterozygous dominant. They inherit a dominant allele. In this case, it was blue, so from dad. And they inherit a recessive allele from mom. All 100% of their offspring. And their phenotype, 100% of their offspring will be tongue rolling. Tongue rollers. So, check that out yourself. See if you can roll your tongue like that. And if you can, you have a dominant allele for it. This actually is a true scenario. And if you don't, you're like me. You did not inherit a dominant allele. You have 2 recessive, and therefore you can't roll your tongue in that fancy shape. So try that at home. And then the last one we're going to try together here is number 4. So in humans, widow's peak is dominant over no widow's peak. Once again, let me go grab an image there. Okay, so widow's peak. Some of you may know what I'm talking about. Your hairline. So it's a simple dominant recessive inheritance in humans. This idea of a widow's peak. Not loving her widow's peak. It's not that profound. Let me see if I can find a better picture for you. So I do not have one, or once again, I'd show you. The widow's peak is this right. Oh, this is a good one. Let's steal this guy right here. Widow's peak is when your hairline has this little V shape in it. Okay, so that's having a widow's peak. Not having a widow's peak is when your hairline just goes straight across the top, like mine, which, there you go. So this is widow's peak. So in humans, widow's peak is dominant over no widow's peak. So once again, you probably already realized I did not inherit a dominant allele for widow's peak because I don't have a widow's peak. Check out yourself at home, see if you do have a widow's peak. So draw a Punnett square to show a cross between a homozygous dominant person with, oh my goodness, I did and, should say, with a widow's peak and a person who, I should have probably spell checked this before I started my video, a person who does not have a widow's peak, okay? So we'll put the person without a widow's peak over here. What is their genotype? Well, if they do not have a widow's peak, they do not have a dominant allele. So they are lowercase p and lowercase p. Now, the parent with a widow's peak is homozygous dominant. So that means they have two dominant alleles for widow's peak. So when we set up this Punnett square, we're going to go ahead and fill in all four boxes again. Allele from the top, allele from the left. 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 So we see now that their offspring, 100% genotype, is that heterozygous dominant. So 100% of their offspring, notice how I'm using percents now. You could also do ratios. You could say 4 capital P lowercase p to 0 capital P to 0 lowercase p. I'm kind of taking the shortcut here. 100% are heterozygous. The phenotypic ratio here, we see that this heterozygous genotype results in 100% of their offspring having a widow's peak. All right, guys, so I hope you enjoyed learning Punnett squares. It is one of my favorite things to teach. I am so bummed that we are not in the classroom learning this together. I'm trying my best to put together some interactive videos. You will have lots of practice with this on Thursday later this week, and then we will, of course, take our FA over it, and you can demonstrate to me that you understand how to set up, complete, and interpret a Punnett square. Take care, everyone. I hope you're enjoying yourself at home. I miss you. Really bummed about the announcement that we're not going back to school this year, and I am, I'm gonna be a little sad about not finishing out the year. So take care. I'll see you all hopefully